Welcome to Boster Bio. Thank you for your interest in our rabbit monoclonal antibody discovery platform. This is C.J. Shah. Boster's microfluidics-based plasma cell discovery technology, PCD, is best suited for therapeutic and diagnostic antibody discovery. In this presentation, I will start with an overview of the technology, provide details on its workflow and timeline, compare this technology with other antibody discovery technologies, and cover some amazing offers for downstream development, such as humanization, antibody pair development, diagnostic kits development, and scale-up production. The main benefit of our platform is its ability to efficiently isolate the small percentage of plasma B cells that produce the best antibodies. With this ability comes high affinity, high specificity, and high diversity of the resulting clones. Before we dive more into why these benefits matter and how our technology achieves them, let me tell you briefly about our company. Boster Bio is an antibody manufacturing company. 30 years ago, in 1993, Boster was founded in China. We are known for making high-quality antibodies and ELISA kits. According to bios.com, our antibodies and ELISA kits have been cited in more than 50,000 publications by 2023. Making great antibodies is our passion. Next, let's go through the PCD technology's workflow and timeline for each step. The total time for producing a rabbit monoclonal antibody using PCD technology is between 6 to 7 months. We typically start with antigen synthesis, which takes 2 weeks. Then, immunization takes 2.5 to 3 months. Next, comes the core of the technology. This phase takes 2 to 4 weeks. By using proprietary chemistry, we can immobilize antibodies on the surface of antibody-secreting B cells. We then incubate extracted splenocytes with fluorescence-conjugated antigens and plasma B cell surface markers, and use FACS sorting to isolate the plasma B cells with the target specificity. There are a few benefits to this approach. Firstly, with our method, the splenocytes are fixed and we can screen for up to 2 weeks without having to keep the cells alive, which allows us to screen through the entire splenocyte population to identify the most rare population of antibody-producing cells that generate the best antibodies. This screening scale is magnitudes larger than any other screening technology. To clarify, on paper, library display technologies have higher screening scale, but if normalized by deducting the percentage of heavy and light chain mismatches, it has an inferior screening scale than PCD. Secondly, by working with plasma cells as starting material, we get to screen only for antibody clones that have gone through gene rearrangement, gene conversion, and somatic maturation. This allows us to take full advantage of the host's natural ability to produce antibodies with high affinity and high specificity, and, in practice, enables us to consistently generate better antibodies than those commercially available made by other technologies. We will illustrate this in a case study later in the video. In the third phase, we clone the heavy and light chains of the positive clones into expression vectors and perform a pilot production of antibodies. This phase takes 4 to 6 weeks. Typically, supernatants from 20 to 40 clones will be provided for the customer to test for the intended application at this stage. If interested, Boster Bio also offers immunoassay services including Western blot, immunohistochemistry, flow cytometry, and ELISA. Quite often customers would contract clone validation work to our CRO division to streamline this process. Please note that the time spent for validating these candidate clones is not calculated into the timeline. Lastly, after determining the candidate clones the customer wants to keep, we will perform clone deconvolution to make sure the clones are stable and free of mutations. Then, we will perform a 30 milliliter media production which typically yields 1 milligram of antibody. Additional clones and additional antibody production can be purchased. This phase takes about 2 weeks. After this phase, the project is complete, and the final deliverable includes at least 1 milligram of the antibody, the validation report, and the sequencing report. Also, 10 nanograms of plasmid containing the heavy chain and light chain genes will be provided, and this plasmid can be used for transient expression of the antibody. Next, let's look at how our PCD technology compare with other antibody discovery technologies. Let's start with an overview of popular antibody discovery technologies. The most classic one is hybridoma, but it is rarely used in high-end antibody discovery due to its limitations. Display technology is another popular antibody discovery technology, and we will focus on comparing our technology with it. Before we dive into this, let me quickly address the key difference between our technology and the single B-cell technology pioneered by Roche. The main difference is in the starting material. We use plasma cells, whereas Roche's single B-cell technology does not pre-enrich its B-cell candidate pool. The first benefit of our technology is the fact that it uses rabbit as a host. Compared to mice, rabbits have a very diverse gene pool of complementary determining regions. This slide shows some major differences between a rabbit's immune system and that of a mouse. Having high CDR diversity leads to higher affinity and specificity. Think of CDR as Lego pieces. The more diverse your Lego collections are, the more complex shapes you can build, and therefore, the more likely one of the shapes will fit a given epitope more tightly, resulting in higher affinity and specificity. Another benefit of higher CDR diversity is more comprehensive epitope coverage. This is important because the more comprehensive the epitope coverage of a polyclonal antibody, the more likely we can select a monoclonal antibody from it that binds to an epitope of interest. This is especially critical in making therapeutic antibodies where the intended application is to neutralize or inactivate the antigen by binding to a functional domain. This is also essential for diagnostic antibodies that intend to differentiate antigens from closely related antigens, such as point mutations, post-translational modifications, and protein cleavage sites. Therefore, using rabbit as the host is more advantageous than mice due to rabbits having a higher CDR diversity. The second benefit of our technology is its screening prowess. Our technology can screen through the entire spleen if needed. Although we typically only screen for 2 days, covering about 10% of the splenocytes, we have plenty of past cases where we successfully produced difficult-to-make antibodies by screening for longer periods of time, up to 1 week. For antibody discovery, our philosophy is to screen as much as possible and as early on as possible. The third point is supplementary to the second point about screening efficiency. When comparing our PCD technology to library display technologies, the main difference is when you use whole cells for screening, the heavy chain light chain pairing is native. When you recombinantly express heavy chain and light chain from a library, the random pairing of these chains result in most of the combinations being nonsensical. Let's assume you have 1,000 heavy chain genes and 1,000 light chain genes. For each heavy chain, only 3 of the 1,000 light chains can effectively pair it to bind to anything. This means each 1,000 screening events with library-based technology only equals to 3 screening events with cell-based technology. These are just hypothetical numbers, but I hope they get the message across. Here is a summary of the pros and cons of our technology. I am by no means claiming that library display technologies are inferior. Rather, our technology can achieve similar results as library-based technologies with much less budget and screening time. However, there is a major drawback of our technology, which is its reliance on immunization of real animals. While this brings much benefit in terms of antibody affinity and specificity, it also causes some issues. First issue is regulations. Some countries are moving towards requiring antibodies to be produced without using animals at all, and this may become the norm in the future. Second issue is in creating antibodies against antigens that have highly homologous orthologs in rabbit. Rabbits may treat the antigen as self and give minimal immune response to the antigen. However, if your project is outside of these two specific scenarios, our PCD technology will be a great fit for you. Let's take a look at a few case studies. These are the results of one of our past projects. As shown in the picture, the 28 isolated clone candidates all have unique sequences. This means our PCD technology has powerful screening capacity and is not affected by immune selection bias. In this analysis, We tested 4 clones generated using PCD technology against commercially available antibodies in SPR. As you can see, all 4 clones significantly outperformed the benchmark antibody. Similar data from many other past projects support that the PCD technology is superior in finding high-affinity antibody clones. Finally, let's find out what downstream services Boster Bio can provide. For diagnostic customers, we have optimized workflows to take a concept to kit. Starting from generating recombinant proteins, producing pairable antibodies, and developing ELISA kits and other antibody pair-based assays. As an ELISA kit manufacturer, we have all the pieces of the puzzle and have developed more than 2,000 ELISA kits. We can also offer preclinical validations for your kits and act as your manufacturing partner if you take the kit to market. To know more about our special workflow for generating pairable antibodies, contact us today to set up a meeting. Other than ELISA kits, we can help validate antibodies to be used in histopathology, flow cytometry, and other formats of diagnostic assays. Check our website for more information about our immunoassay services. If you need large amounts of your antibody, we can produce a bioreactor-based antibody production service. We can generate up to gram levels of antibodies. Finally, if you need to humanize your antibody, we have partners who specialize in antibody engineering and antibody humanization. Schedule a meeting with us for more information. Thank you for watching. Feel free to let us know if you have any questions.